Now that we are incorporating PD-L1 antibodies in the first-line setting, atezolizumab or durvalumab with chemotherapy, for those patients that don't derive that long-term benefit, what are our second-line options? Our approved option, our standard of care here, is topotecan, a drug that's FDA approved, but a drug that we're not too excited about. Fairly toxic, not particularly effective. The response is fairly transient. And so what are the other options? There certainly is an opportunity, an unmet need to develop new drugs in that space. And one of the more promising drugs, probably closest to regulatory approval, is lurbinectedin. Lurbinectedin is a marine-derived molecule that has a direct anti-tumor effect, affecting DNA transcription, and it's shown promising activity, both as a single agent and in combination with doxorubicin. And so this is a drug that we hope to be able to make available to our patients relatively soon. It is not without toxicity, primarily hematologic in nature, but often those are paper toxicities that we can manage that don't necessarily decrease the quality of life for specific patients. So the activity we've seen has been promising, but we're waiting for randomized data to really show us the precise benefit of lurbinectedin. And there are several other drugs in development. One, unfortunately, drug that's been taken out of development is Rova-T. And so this, a drug targeting DLL3, had shown promising activity initially. Unfortunately, serial trials since then have shown disappointing outcomes, particularly with regard to safety and toxicity. At ESMO 2019, we saw one of the first-line trials with Rova-T in combination with chemotherapy being studied, and unfortunately that trial did not meet its primary endpoint, and so that drug will not be further developed. It does not negate DLL3 as a possible target, and because this is expressed on small cell lung cancer, not normally expressed on other healthy tissue in adult patients, it still is a viable target that can be exploited. It still represents this context of vulnerability for small cell lung cancer, but perhaps a different strategy. And so there are BiTEs, there are CAR T cells targeting DLL3 that are in development now, and hopefully will be able to be added to our armamentarium in the near future. I think that for extensive stage disease, we're still looking for novel agents, for better options for salvage therapy. But there are sort of two main areas of research that I think will have the biggest impact. First is in limited stage small cell lung cancer. About 1/3 of our patients will present with limited stage, a potentially curable stage. With a combination of chemotherapy and definitive thoracic radiation, we will cure some patients, but not most patients, probably somewhere around 1/3 at best of those patients. So how do we increase the rate of cure there? And we're looking at incorporating immunotherapy in limited stage in combination with chemotherapy and radiation, building on our experience with immunotherapy in Empower 133 and Caspian, and building our experience in combining immunotherapy with radiation in Pacific, non-small cell lung cancer. And so we have the Adriatic study, looking at patients who'd received chemotherapy with radiation for limited stage small cell lung cancer, who would then receive durvalumab consolidation. And we have NRG-LU005, an NCI cooperative group study, looking at chemotherapy with radiation and concurrent atezolizumab given at the same time as radiation. And so these two limited stage studies will hopefully lead to more cures and prevent relapse for a significant portion of our patients. Really, the last frontier is biomarker work. And what we're seeing with these immunotherapy studies with Empower 133, with Caspian, is there is a benefit. It's a real benefit, a survival benefit, but it's being carried by a subset of patients. And if we can identify who those patients are, we can be sure they receive the right therapy. And for the patients who aren't in that subgroup, we can explore novel strategies, figure out why they aren't deriving that response, alter our treatment approach so that it can develop that immune response, either with addition of things like PARP inhibitors, radiation, other novel checkpoints, drugs targeting the microenvironment. And so it comes down to appreciating the differences between small cell lung cancer and some of the newer work being done in terms of transcription expression to identify specific molecular subsets of small cell lung cancer will be very important in taking that next big step forward. One thing to keep in mind with extensive stage small cell lung cancer, it's a very rapid pace of disease. This is an aggressive cancer that moves quickly and will not tolerate delays. When we look at some of the early placebo-controlled trials in the 1970s, we see the natural history of this disease is really a survival limited to a few months. And so it is an unforgiving cancer. Delays will really not be tolerated and may lead to patients losing that opportunity to receive a therapy that extends survival and improve quality of life. And so if you wait for conditions to be perfect, you might be waiting too long. So a lot of the patients we treat for small cell lung cancer don't fit that clinical trial population. They have a worse performance status, more symptoms, perhaps untreated asymptomatic brain metastases, and yet we need to move forward, because if we wait for everything to be perfect to meet trial conditions, then that patient, again, will lose the opportunity to really receive very beneficial therapy. And that therapy has been chemotherapy. Platinum etoposide, first introduced in the 1970s, really our standard of care since the 80s and 90s. And what chemotherapy does is it induces responses. They can be fairly quick, they can be deep, you can have complete responses. Nearly all patients will feel better, but that benefit is transient, and progression-free survival usually around 4 or 5 months. So patients progressing shortly after or during chemotherapy. And when cancer relapses, when small cell lung cancer recurs after platinum etoposide, it's highly refractory to the treatments that we have. And so it's important to maximize the benefit in the frontline setting. And we've looked at different variations on platinum etoposide. We've looked at cisplatin versus carboplatin. We've looked at 4 cycles versus 6 cycles. We've seen different doses of platinum, of etoposide, split doses. All these variations didn't really make much impact. And if there's any difference, it's incremental at best. And these were the debates we had for years because we had nothing else to talk about. And now finally we're starting to make some advances.